La vediamo. Good morning, everyone.
Good
Good morning, sir.
Has everyone joined?
Yes, sir.
Okay, can we start?
Yes, sir.
Okay, so let us start. Welcome to today's session. Today's session is about microbiological analysis. Before the start of our session, I would like to introduce our today's speaker, Dr. Pushpa H. And Dr. Pushpa H is a distinguished microbiology professor and vice principal at MS Ramaiah College of Arts, Science and Commerce with a PhD from Bengaluru University. She's a leading expert in fungi taxonomy as well as biodiversity. As the DBT Star College Coordinator, she's instrumental in administration, research, and contributed more 20 plus research publications, international collaborations, and patent filings, and also recognized with awards like Emerging Leader and the Most Fabulous Professor Award. And we welcome you for today's session, ma'am.
Thank you, Vishal, for your briefing about me. Thank you so much for that. Good morning, students. Can you all hear me?
Yes,
Good
ma'am.
morning, ma'am.
Yes,
Yes,
ma'am.
ma'am.
Good morning, ma'am.
Good morning. So today's session is with respect to this microbiological analysis. So, so all these days, especially in this microbiological unit, you would have studied about the various microbiological aspects. So today I'm going to talk about how actually we are going to analyze the microorganisms in the sterile manufacturing. So today I'm going to highlight. So here I'm going to highlight about what actually is this sterility test, why it has to be performed, and what actually why this sterility test for pharmaceutical products are required. What are the environmental conditions required for this sterility testing, and how we are going to monitor this environmental condition methods and the techniques. And I'm going to talk about the different products of pharmaceutical products, how the sterility testing is being done, what are the different steps involved in the sterility testing. And I'm going to talk about how many sampling and how the samplings has to be done as per the USB, that is according to USB 71. And I'm going to talk about the different culture media which is required in this particular sterility testing process, which are the different, what are the composition of this particular media, and how this media validation has to be done, and which are the microorganisms which we are going to use in according to USB 71, and the two important methods which we are going to follow in the sterility testing. One is membrane filtration method, and the other is direct inoculation, and how we are going to interpret the results. So these are the things which I'm going to highlight in this 2 hours class. So first, what actually, if we are going to call this particular unit as microbiological analysis, the main outcome, what we are understanding in this particular unit is how actually we are going to sample the pharmaceutical products, which are the different criteria we have to use according to the US Pharmacopoeia at 71, and then how actually this technique is been done and how we are going to interpret the result. That is what the outcome of this particular unit. First of all, first, I would like to give an introduction about this sterility testing. We all know what is the meaning of the sterile condition. Can anybody let me know what is the meaning of the sterilization? I think most we are studying sterile manufacturing itself. Can somebody tell me about the meaning of the sterilization? What is sterilization? What is sterility? Yes, can anybody tell? Joshua Johnson?
Sterilization is a
Mm.
process of eliminating the
Yes.
um, disease-causing organism, no?
It need not have to be disease-causing organism. Any of the microorganisms is the process of eliminating the microorganism in the given sample. That is called as sterilization. And also, we all know the sterilization can be done by various processes. There are, can anybody tell me, Aditi, can you let me know what are the different methods of sterilization? Sterilization, we know it is the process of elimination of the microorganism in the given sample. Okay, what are the different methods of sterilization, Aditi Pandey?
Yes, ma'am.
Yes.
Sterilization can be done by different methods like dry heat sterilization, moist heat sterilization,
Very good.
by
Yes.
chemical method, by
Very
radiation method,
good.
and by
Yes.
filtration.
Yes. So the sterilization can be either a physical method or it could be a chemical method can be used. The physical method, we all know the different methods of physical. We have the heating method, we have the radiation method, etc. So here, my thing here is how we are, here the thing is whether the pharmaceutical products. So what are these, if I am going to call it as a pharmaceutical products, what are the different pharmaceutical products? What could be the different pharmaceutical products? Yes, it could be injections, it could be some syringes, bandages, dressing materials, it could be the needles, or it could be injectables, or any kind any kind of the liquid solutions or the solid formulations. They are the products of they are the pharmaceutical products. So if you are going to call them as a pharmaceutical products, the main purpose here is with respect to the health reason. We are going to use this thing when we are not okay, isn't it? So if these pharmaceutical products itself is not sterile, so how actually, I mean, what is the purpose of giving this? So that is the reason this pharmaceutical products should be sterile. So here, in order to see whether these particular products are sterile or not, we have to conduct this sterility testing. Okay, so the sterility test, that is what we are going to discuss in this particular class. The sterility test is a test that critically assess whether the sterilized pharmaceutical product is free from the contaminating microorganism. So here we are going to perform the test to see whether the pharmaceutical product, as I had listed, whichever the pharmaceutical product it could be, either it could be an ointments or injectables or tablets or dressing materials or syringes, whatever it is. Here we have to see whether they are free from the microorganisms or not. So that is the reason we have to conduct this sterility test. So according to Indian Pharmacopoeia, the sterility testings are intended to detect the presence of viable forms of microorganisms or on pharmacopoeial preparations. So that is viable form of microorganism. What is the meaning of viable form of microorganism? Deepika, can you let me know what is the meaning of viable form of microorganisms? So here, according to Indian Pharmacopoeia, the sterility testing are intended to detect the presence of viable form of microorganisms. What does it mean? Viable form of microorganisms.
Ma'am, the living ones which can reproduce, ma'am.
Yes, very nice. So here we would like to analyze whether the product, whatever the pharmaceutical product, whether it is having the microorganism which are alive. So that is the meaning of the sterility testing. Then apart from having this living organism, this living organisms or the latent organisms, they also have an ability of producing the substance or an endotoxins called as pyrogens. These pyrogens are a group of chemically devised substances that cause fever and shock in severe cases. The most important pyrogenic substance in pharmaceutical industry and are the bacterial endotoxins. Bacterial endotoxins are the part of the cell wall of gram-negative bacteria. So here, one thing is during the process of sterility testing, we are going to analyze whether the living organisms, the contaminant or the microorganism there in that particular product, and at the same time whether they have any endotoxins or pyrogens. What are these endotoxins? These endotoxins are nothing but the substance, or especially the lipopolysaccharide, which is being present especially in the gram-negative bacteria. So here I have given the chart showing the difference between the exotoxin and the endotoxins. So exotoxins you could see here in this particular okay. So here you could see the difference between the exotoxin and the endotoxin. Or this exotoxins are the proteins produced inside the pathogenic bacteria, most probably, most commonly, gram-positive bacteria during their growth and metabolism. They are produced inside and excreted outside, and they are released during the process of their lysis. Lysis in the sense what? The breaking down of this particular cell wall and the cell membrane. So those are called as exotoxins. So here the pyrogens are the substance which are found to be toxic, and they are the endotoxins which are produced especially the gram-negative bacteria. So can anybody let me know the difference between the gram-positive and gram-negative bacteria? Shubhadeep? Shubhadeep?
Yes, ma'am.
Yes, ma'am.
Gram-positive bacteria, peptidoglycan layer is thick,
Very
and
good.
gram-negative bacteria, peptidoglycan is thin.
Yes. And apart from
And
that, something gram-negative bacteria has. Lipopolysaccharides. Lipopolysaccharides. Those lipopolysaccharides which are present on the surface of the, on the cell wall of this gram-negative bacteria, they are the one which are liberated when they die, when the cell wall break up. Those are called as endotoxins. These are also called as pyrogens. These pyrogens are the one which causes the various diseases, or especially the fever, especially because of this particular substance. If it is present in that particular pharmaceutical product, if gram-negative organisms are dominating in that particular product, they have an ability of producing these pyrogens. If this pyrogens has been used by any of the patients, they causes fever and shock in severe cases. So that is also the reason we have to analyze the particular product is sterile or not. So some of the examples of this pyrogen producing gram-negative bacteria has been listed out, especially the dominant group of organisms which has the potential of contamination are the E. coli, Proteus, Pseudomonas, Enterobacter, Klebsiella, etc. So how actually we can analyze this particular endotoxin is present in the given sample or not? So there are two different methods. There are two different methods in which we can detect the presence of this endotoxin. One is rabbit pyrogen test. That is where the rapid pyrogen test is based on rectal temperature of the rabbit. That is measuring the temperature before and after the 4 injection of the test solution. So you could understand it very easily. That is the temperature, that is the rectal temperature of the rabbit increases if the test sample, if we are injecting into the rabbit and the temperature increases. Why the temperature increases? If the pyrogens are present in that particular sample, pyrogens in the sense what? They are the lipid polysaccharides. Where you can find this lipid polysaccharide? If there is any contamination of gram-negative bacteria, which are the, if such gram-negative bacteria, if it is present in that particular sample, obviously the endotoxins can also be detected. The detection of this endotoxin can be done. One of the method is by what? Rapid pyrogen test. That is, we are going to inject this particular rabbit with the test sample. If the temperature increases, indicates the chances of having this pyrogen in that particular sample is more. The another important test is called as LAL test. LAL, or it is also called as limbless amoebocyte lysate testing. It is because the lysate from the blood cells from horseshoe crab. The Latin name is limbless polyvirus. This is from there. The lysate is being taken, and this particular lysate is mixed or allowed to react with the test sample. If there is any change in the color over a period of time, indicates the presence of this endotoxins. Okay, so this is also one of the method. One is LAL test, the other one is rabbit pyrogen test. That is the lysate from the horseshoe crab that we are going to that thing is being mixed with our the test sample. If there is any color change that is yellow over a period of time, indicates the amount of, if the concentration or the color intensity of the color also indicates the concentration of the endotoxin present in that particular sample. So here we could analyze the two different today. I mean, in this particular slide, we could analyze that why the sterility testing has to be done for pharmaceutical product in order to see whether the pharmaceutical product is devoid of this particular microorganisms, and this can be detected. One is by directly testing whether the microorganism present or not. The other method is the endotoxin if they are producing by this organism. Endotoxins are usually produced by gram-negative bacteria, that is the lipid portion, that is the lipopolysaccharide present in the outer layer of the cell wall of the gram-negative bacteria. They are the endotoxins which are released when they die and the cell wall breaks apart. Okay, so this can be detected by two methods. One is rapid pyrogen test, the other one is LAL test. That is what we could see in this particular slide. Next, let us go for the microbiological, and our target is to see the microorganism in the given sample. So what are, I mean, how actually we can do this and why we have to do this particular testing? So the main purpose of performing the sterility test for this pharmaceutical products are to reveal the presence of viable forms of bacteria, molds, and the yeast in the pharmaceutical products or devices. Once again, I will let you know here the main purpose of conducting the sterility test is to find out the presence of viable form of bacteria, molds, and yeast. So here it is not only the bacteria, we should also analyze the presence of molds. What are these molds? Shashank, what are molds?
Molds are the spores-like formation.
Molds, spores, correct. Can you redefine from where you will get the spores? Spores.
It is an inactive form of bacteria.
No. Molds. What are molds? Lasya?
Ma'am, fungus, fungal
Fungus.
spores.
Molds, they are the fungi. The fungi, you can characterize them based on their appearance as molds, yeast, and mushrooms, isn't it? isn't it? So then I should ask Neeraja, can you let me know what are these fungi then? So molds, some of them are telling they are spores like thing, and some of them are telling they are fungi. Before that, what are these fungi? What are fungi? Irene?
Yes, ma'am. It is
Yes.
a eukaryotic organism.
Many are eukaryotic organism.
It is a microorganisms like yeast.
No, that is what they have been, all these molds, yeast, mushrooms have been placed under the kingdom. In which kingdom they have been placed? I mean, you know, you have the five kingdom.
Fungi, kingdom fungi.
Kingdom fungi. Why they have been placed separate?
I don't know, ma'am.
Can anybody can tell me the five different kingdom?
Kingdom prokaryote.
No.
No. Monera, Protista,
Monera?
fungi,
Yes.
Plantae, Animalia.
Very nice. Very good. Then on what basis it has been classified? On what basis?
Monera is the prokaryotes,
Very good.
unicellular prokaryotes.
All so you can place bacteria. Can you place bacteria in Monera?
No.
No.
They are prokaryotes, Monera, unicellular, and Protista is unicellular eukaryotes. Fungi are multicellular. Plantae.
So this will be an assignment after. I will give you a break later. So you have to, I think, then we shall have a discussion on this, the five system of the kingdom of the class, according to Whittaker system of classification. We have five kingdom. On what basis they have been classified, that we shall have a discussion. Fine. So now I'm going to tell you all these fungi have been placed as a separate kingdom because the main reason is they are eukaryotic, they are achlorophyllous. Achlorophyllous in the sense they doesn't have the chlorophyll pigment in them. Because of that, they are not the producer. So they are dependent on the other organisms, especially they are saprophytes where they grow on dead and decay matters of plants and animal remain. They are basically the saprophytes. Not only the saprophytes, they can be a symbionts, they can be also a parasites also. Okay, so I for talking about this fungi itself, I may take many classes. So just one definition, the fungi or the achlorophyll, they are eukaryotic, achlorophyllous, spore-bearing, saprophytic organism. Okay, these keywords you just remember now. These fungi, based on their morphology, you can classify them as molds, yeast, and mushrooms. What are these yeast? Yeast or unicellular fungi. Yeast-like organism, unicellular fungi. Molds, they are having a mycelia-like structures. Example, I think you would have seen, have you all seen Rhizopus, Mucor, Aspergillus, Penicillium? Have you all seen them? Have you all seen them? Deepika? Have you observed yeast? Sorry, have you seen yeast under microscope? No? Deepika, HB?
Yes, ma'am.
Have you seen yeast?
Ma'am, the one like we did grow in the lab in I mean on the bread, what we found, we did observe that on the in the microscope, ma'am, under microscope.
Yeast, how it was? What was the morphology of that yeast?
Filamentous, ma'am.
Yeast, or I'm telling that yeast or unicellular fungi, isn't it?
It is filamentous.
So here in this particular program, skills are more important than the theory, isn't it? Isn't it? Hands-on experience, the skills are important. So if I give you the sample and if I ask you to identify whether the yeast is there or not, first you should know what these yeast are and how actually it looks, isn't it? Have you observed penicillium, any molds? Molds in the sense the fungi which are in the form of hyphae or a thread-like structures and producing spores. Have you seen Penicillium, Aspergillus, Rhizopus?
Small.
Okay, those are molds. I think even in your UG classes, you would have observed them, isn't it? Then mushrooms, they are macroscopic. These molds and yeast are microscopic. To see their morphology, you require a microscope. Whereas these mushrooms, they are macroscopic in nature. So here, let us come back to our topic. Here, our aim is to see the presence of viable form of bacteria, whether the sample has got bacteria or a mold or the yeast or yeast in the pharmaceutical products or devices. To monitor the quality of the pharmaceutical product before marketing. That is, before you are going to market that particular pharmaceutical product, we have to analyze and monitor whether whatever the product we are marketing is devoid of this particular microorganism. To avoid the contamination, to assure the safety of the product, isn't it? We have to assure, yes, our product is free from the microorganism. It has crossed all the QC. So that is the reason we have to perform this sterility test. One thing, to see whether our product has the viable form, that is the living form of this bacteria, that is they can live or they can reproduce. So such kind of organism is there or not, we have to analyze, and to monitor the pharmaceutical product before marketing to avoid the contamination. Contamination in the sense the microbes, if they are there, they are called as contaminant, and to assure whether the product is safe or not. So that is the reason we have to go for this sterility testing. Okay. Before going for sterility testing, if I'm going to, you just imagine that we are going to give some lot of the pharmaceutical product, and we are asking you to analyze whether they are devoid of this microorganism or not. So before going to that, the place where you are working, that is the environment where this particular sterility testing is being done or not, is sterile or not, that we have to analyze. So that is sterility testing environment. The test area must be maintained to minimum the possibility of contamination of the test. Any measurement taken to prevent contamination from the test must not affect the microorganism. The test area should be adapted to create stable, easily monitored and controlled condition, that is the aseptic condition. Testing laboratory should perform the environmental monitoring to demonstrate the control of the test area and provide the indication of failure in control. Sterility test should be carried out by trained technician. That is, the environment, the laboratory in which we are working, it should be maintained sterile, or else the results, whatever we are getting the results, because if the microbes themselves, if the environment, the area where you're working itself is not clean, if they are not devoid of the microorganism, the result interpretation may go wrong. So that is the reason the testing area where we are working, it should be maintained sterile. How do we maintain this particular environment sterile? How do we monitor this environment or the working region sterile? How we are going to do this? So here the environmental monitoring system is to demonstrate, that is, the living organism or non-viable particles in a critical area. These areas includes, which are the areas that the region where we are working. It includes the clean room, or it could be a formulation tank room, laminar airflow hood, biological safety hoods, isolators, gloves, molding machines, kits assembly linings, etc. So here, these are the region where actually we are working, especially with this pharmaceutical products. These environment, especially for the sterility testing. So these environment, we have to maintain, we have to monitor these environment. How we are going to monitor these environment and which are the things we have to monitor is, one is one thing is the clean room, that is the area where we are working, that is called as clean room. So how we are going to monitor this clean room? One is the person or the personnel who are going inside this particular clean room. We have to monitor the personal hygiene or the personal person whoever is entering in that particular room. We have to monitor that particular person, the different wears he is going to wear or she is going to wear. It is sterile. How we are going to do that, I am going to let you know. Apart from this personnel, we have to monitor the air, that is the environment, that is the air microflora of that particular clean room has to be maintained. Another thing is we have to also maintain, we have to also see the surface of that particular room. It could be a floor or it could be a wall or a equipment, whatever there in that particular room is clean or not. So these are the things which we have to look in to see the environmental monitoring. One is, the first thing is the personnel. Personnel are the biggest source of contamination in the clean room. That is the person, whoever is entering in that particular room, there are chances of carrying various, many or millions of microorganisms wherever they go. And this is the one of the chances of contamination of this particular room. That is the reason the personal monitoring employs the contact plates to assess the microbial monitoring of the clean room. That is, what we have to do is, whatever the gloves he or she is wearing or the gowns they are wearing, the mask or whatever they are wearing, they are sterile or not. So in order to see that, the microbial examination has to be done. How this microbial examination has been done is, one method is by contact method, contact plate method. Other one is by taking the swabs. So here, whatever the wears or the gloves or gowns or whatever it is, the media has to be prepared, appropriate media for bacteria or fungi. It has to be prepared, and the samples has to be just baited, or the sample has to be made to come in contact with that particular surface of the agar, and the plates are incubated and see for the growth of this particular organisms. So that is one method. Other one is swabbing. That is, you are going to collect the swabs in the sense sterile swabs has to be taken. It has to be rubbed onto the surface of the gowns or whatever it is, and they are inoculated onto the surface of the culture media, incubated, and see for the growth of the organisms. So that is how we have to monitor the presence of the bacteria or a fungi in that particular sample. The other one is air. Air is also another important thing in the environmental monitoring. Air in the clean room is controlled and monitored on regular basis. On regular basis, that particular clean room in the sense, the room in which we are performing this sterility testing, please keep in mind. That clean room, we have to monitor the quality of the air. Quality of the air in the sense, especially the microflora, the temperature and the humidity of that particular room has to be maintained on every daily basis, weekly basis, or quarterly basis. That has to be maintained. How we are going to monitor this particular air of that particular clean room? One is air samplers, using air samplers. Air samplers, it can be of two kinds. One is active air sampling and the passive air sampling. active air sampling and passive air sampling. In this active air samplings, we are going to use such kind of samplers. We are going to use the samplers. Air samplers here it will, this particular sampler have an ability of sucking in the air in that particular environment. And also we should also note that here the media is being placed in this particular sampler. Here, once the air is been sucked by this particular sample, so in this particular air, if the microorganisms are there, they are going to settle on the surface of this particular media. Once it has happened, once we are going to incubate such kind of plates, we could see if there are viable organisms, you could see the growth of this particular organism. So that is called as active air sampling. That is, we are going to use the samplers. The samplers can be, here in industries, we can, the impact method they are going to use. One is, we have a bench top air sampler. This is one of the image which I have put, the bench top air sampler, that is handheld air sampler. That is, these are the kind of air sampler where we can hold this particular air sampler, go to a particular place, and we can suck that particular, this particular samplers can suck this particular air. A particular time also can be monitored, and also the once we monitor the time, what happens, this particular microorganisms which are there in that particular air, they just come and land on this particular media which is there inside. And once they have been landed at that particular point of time, we can take out that particular plate and incubate, and you could see the microorganism. So here we are going to provide a plate containing the media. For a bacteria, the media are different. The fungi, the media are different. If you want to see which fungi is there, which bacteria is there, prepare appropriate media, sterilize the media and place it in this, and suck the air using this sampler. And once the microorganisms are adhered on the surface of this particular media, take out this particular media and incubate at appropriate temperature. For bacteria, the temperature is different. Fungi, the temperature is different. Incubation time and incubation period is different for bacteria and fungi. Maintain that. Once the incubation period is over, take out those plates and observe for the growth of the microorganism. Calculate the number of colonies or the CFUs present in that particular plate. That is called as active air sampling. In case of passive air sampling, here the samplers are not required. What we can do is the Petri plates containing the sterile growth media. Growth media, once again, I will tell, for bacteria and fungi, they are different. Prepare appropriate media, pour the media, sterilize the media, pour it onto the sterile Petri plate, allow it to solidify, and place such kind of Petri plate, open the plate and keep it in the clean room for about, it depends upon the number of time also, 30 to 60 minutes. And you can plate it, expose it for 30 to 60 minutes. And also it depends, time also depends, 5 minutes, 10 minutes, 15 minutes, etc. Then once the exposure time is over, close the lid, keep it for incubation at appropriate temperature and time, and monitor for the growth of the organism, and calculate the CFU and interpret the results. So this is how the passive air sampling is being done. So one is by using samplers, one is by using air samplers, the other one is by just by settle plate method, preparing a nutrient agar or a required media for fungi, plate it on a Petri plate and expose it, incubate, and see for the growth of the viable microorganisms. This is how we are going to monitor the air in that particular clean room. And another thing is the surface of that particular clean room. Clean room, once again, I will tell, in the sense, the place or the room or the area where we are going to perform this sterility test. That is your clean room. So one thing is the personal. The second one is air, whether the air is devoid of microorganism. Third, whether the surface of that particular room, which includes floor, walls, equipments, whichever the region it is, whether it is clean or not. So for that also, prepare appropriate growth media or nutrient agar or triplicate soy agar or sabardol dextrose agar for fungi, etc. You can go for contact plate method or you can go for swabs method. Contact plate method, that is the petri plate, petri dishes which are containing the sterile medium. We are going to whatever the surface, no, we are going to bring that particular surface with the contact of this particular media, and then we are going to incubate. That is called as contact plate method. Swabs, the sterile swabs, we are going to have a sterile swabs. These sterile swabs, we are going to rub it onto the surface of the floor or equipment or whichever the region. We are going to collect that particular swab, and then we are going to inoculate that particular or streak or inoculate that or swab that particular thing on the surface of the solidified culture media, and we are going to incubate and see for the growth of the microorganisms. And if possible, you can subculture those organisms and identify them till genus and species level. So this is how we are going to monitor the environment of the manufacturing or the clean room. One is personal, that is the person whom they are entering inside, the wares they are wearing, they are sterile or not, that we have to monitor. The chances of contamination is majorly through that. That is the reason the person should be, the wares they are wearing should be sterile, and the environment, that is the air in that particular region, should also be maintained sterile. And also this thing can be done by using samplers. It could be a active air sampling or it could be a passive air sampling, a simple method, settle plate method we are going to follow. The surface also in that particular, whichever the surface in that particular room also has to be monitored in frequent intervals of time. We can go for a contact method or the swabs method. So this is about the room which we are going to monitor. Next. Next thing is our aim. Now, the working area, we are going to, we have monitored whether they are sterile or not. Next thing is, which are the products can be analyzed for sterility testing. As I was mentioning in my previous slides also, the pharmaceutical products, few examples, many are there, only few examples I have quoted here. It could be injections, implants, syringes, bandages, dressing materials, dressings, surgical equipments, needles, injectables, bulk solids, or the ophthalmic products. These are the some of few examples I have given here. Some of the pharmaceutical products, once from the pharmaceutical company, once in that company, once the production is happened, here it has to pass through the various validation has to be done, quality control has to be done. During that, one of the thing is the microbiological analysis. Microbiological analysis, especially we are going to see whether these particular products are devoid of viable organisms or not. So in order to do the sterility testing, these are the different steps involved in sterility testing. One is the selection of the sample size, selection of the quantity of the product, media requirements, test microbes, methods of testing, and finally observation and results. The first thing is we have to select the sample size. I mean, my next slide, I am going to talk about how many samples has to, what is the size, what should be the size of our samples, how we have to select the sample size. According to ESP, I am going to talk about, and how the selection of the quality of the quantity of the product, how many samples has to be selected. how much quantity of the product has to be selected, which are the different media we have to use for analyzing them, which are the test microbes we have to analyze, and also for validating the media and the different methods of testing. And finally, what actually we have to observe and how we have to interpret the results. This we are going to look in. I think you can take for around 5 or 10 minutes break, and then we are going to come back. So one assignment I have given student thing is the 5 kingdom of classification. What are the different, on what basis they have been classified, that you have to just let us know. Let us have the discussion on that, and later let us proceed with these topics. You can take around 10 minutes break and we will come back again. Hello, good morning, students. Can we all resume back? So everybody is back. Why should
Ma'am,
why-
few are there to come, ma'am.
I had given only 10 minutes break.
Yes, ma'am, they're coming. They'll join now. Ma'am,
Yes, ma'am.
everyone join, ma'am.
Okay, okay, thank you. Dr. Basaya is there? He's absent.
Ma'am, he is employee, ma'am.
Okay. Omkar, kindly let me know how do we monitor the environment in the clean room? How do you monitor the sterile condition in the clean room? How do you monitor? Omkar. Yes, no talking.
Yes, ma'am.
Yes. How do you monitor? So I will give you the some pharmaceutical products, and I will ask you to see for the sterile condition of that particular product. The first thing what we have to do is the working environment should be clean. devoid of microorganisms, or else there is a chances of getting the interpretation of results will go wrong. So that is the reason the room has to be maintained. So how do you do that? How do you monitor that? First, what you will do? So your
By
theoretical knowledge is rather than your hands-on. You just imagine how you are going to monitor that. Yes, you do not have to stick on to the technical thing. You can tell in your own words, no issues.
ma'am, by using contact plates.
What are the things you are going to monitor first? Clean room. I will ask you to enter into the clean room, and I will ask you to see, you give me a report of that particular room, perform the microbiological assay of that particular room, and let me know the results, what you are going to do.
First, I will check the personal,
Hmm.
then the air quality of the clean room,
Yes.
then the surface
Hmm.
monitoring.
Okay. Surface one. And also in your previous classes, you have learned about disinfection, all those, I think, air sampling, everything is being, I think in microbiology, you would have studied all those, isn't it? So the air sampling can be done using samplers as well as by settle plate method. What is the meaning of this settle plate method? Omkar.
Which contain culture media that is exposed by the air at the given time.
At the given time, you are going to place the Petri plate containing the appropriate culture media. And that is, so what happens, as the name indicates, settle plate, microorganism, the microflora present in that particular year, they try to settle by gravity method onto the surface of the media that you are going to, that plate you are going to incubate and see for the colony forming unit later, isn't it?
Yes,
Is
ma'am.
it? That is settle plate method. Okay. Okay, so now in before this break, we studied what is the meaning of the sterility testing, and why we have to do this sterility testing with respect to this microorganism. In order to perform this, how we have to maintain the environment or the place we are working, why we have to maintain that, how we have to maintain the environment, we have studied. And also we studied the different products, the pharmaceutical products, which we can see the sterility testing. Now, let us see how we are going to perform this sterility testing. First thing is, as I was mentioning, selection of the sample size. See, if the pharmacy, lots of some injections or any of the tablets or capsules or gels or ointments or whatever it is, usually the pharmaceutical company will prepare them in bulk. Can we analyze each of the sample? Each of the sample can we test and see whether they are devoid of microorganism? No. So for that, they, according to USP, there are standard procedures which we can follow to analyze to select the number of articles in that particular batch. Students, kindly note, this is particular table which shows the minimum number of articles to be, articles in the sense the samples, minimum number of articles to be tested in relation to the number of articles in the batch. Please make a note of this particular table. Later, I think in your quiz, I may ask few questions out of these. So, and also I may ask a few of the calculations. So you should be knowing, say for example, if you get some batch of some products of pharmaceutical products, out of that particular batch, how many samples should I have to check? How many samples? You cannot check the entire batch, entire or one vial or two vials. It is not like that. According to USP, in the sense, what is USP? What is USP? Anybody can tell me?
United States Pharmacopoeia.
Yeah. According to USP 71, here 71 USP speak about the sterility testing. So according to that, this is the table which they have mentioned. According to this USP 71, the sample size has to be, the number of samples or the minimum samples has to be selected in this way. So here in this particular table, we can see the number of items in the batch and the minimum number of items or the samples has to be tested for each of the media. Okay, so this is what the table is. So here the table represents the parenteral preparation. What is the meaning of parenteral preparations? Anybody? What is this parenteral preparation?
It's a parenteral drug, ma'am, which cannot be taken orally.
Orally, it could be in the form of, it could be in the form of solutions or emulsions, that is injections, infusions. It could be powder, injections, or it could be gels. Especially these are the preparation, these are the sterile preparation which has been administered directly to the systemic circulation in the human or animal body. Okay, so that is whatever the answer which I received, it was correct. That is, which cannot be administered directly orally. That is called as parenteral preparation. This includes usually the injectables. So here, if you are getting some batch of such kind of parenteral preparation, how we are going to do this particular sampling? So for example, the parenteral preparation, the number, for example, not more than 100 containers, if that particular batch, if it is not more than 100 containers, 10% or 4 containers, whichever is greater has to be selected. If you are getting some batch of this kind of preparation, where they contain less than 100 containers, then what you have to do? The 10% of that or 4 containers has to be taken and it has to be subjected for analysis. Likewise, if it is more than 100, not more than 500 containers, the 10 containers. More than 500 containers, 2% of the containers, whichever is lesser. Larger volume of such kind of parenteral preparation, 2% or 10 container, whichever is lesser. This is according to USP. Though here we are, once again, I am going to repeat. So here we are, the main aim of studying this particular table is to select the minimum number of sample. Why we are selecting? To analyze the microbes present in that particular sample. So if that particular batch has got 100 or more than 100 or more than 500 or very large amount, this is what according to USP, this is the number of the articles or number of samples which has to be selected and it has to go for analysis. Likewise, antibiotic solutions also they have given in that pharmaceutical bulk packages. If it is more than 5 grams, 20 containers, less than that, 6 containers, bulk blends. So likewise, they have given there is some standard where the number of the samples has to be taken for analysis. So this is the table which you have to always refer and you have to keep in mind for the process of analysis. So, kindly note down this particular chart. And likewise, if you have this ophthalmic other non-injectable preparations, what is the meaning of ophthalmic? What is the meaning of ophthalmic?
For
Of
eyes.
eyes,
For eyes medicine.
eyes medicine, especially it could be eye drops or the solutions or suspensions or any such kind of ointments. That is called as ophthalmic. And here also not more than 200 containers, 5% or 2 containers. This is the standard. Always we have to know. Likewise, you have catgut. What is the meaning of catgut? Catgut or surgical sutures. The catgut is nothing but the surgical threads. You would have seen the surgical threads in order if you have any during the process of operation or if you have some cuts or something, they will use those surgical threads. Those are called as catguts. So that also the analysis depends upon the number of packages. 2% or 5 packages has to be or greater than that has to be analyzed. So this is the table which represent that you have to remember, and you have to I think you can during the process you have to remember, you can refer, and you have to keep always in mind this is the sampling, this is how it has to be done according to USP 71. Next, minimum quantity, that is the minimum number. The previous table was out of the batch, bulk products, whatever you get, minimum number of samples which has to be selected. Here, minimum quantity, how much amount of the sample has to be collected. That is what this table represents. Minimum quantity of the sample to be used for each of the media according to USP 71. So if it is liquid, less than, for example, imagine ampule or whatever it is, if it is less than 1 ml, the whole content of each container has to be used. If that particular liquid, if it is the sample I'm talking about, now you would have selected sample, 2% or 4 samples or 5% or 2 samples, whatever you would have collected. Out of that particular sample, how much amount has to be used for the process of inoculation. That is, for that also there is some USP protocol has to be followed. If that particular container contain less than 1 ml of the solution or whatever it is, the whole content of the container has to be used. This also you should keep it in mind. If it is 1 to 40 ml, if that particular container has got 1 to 40 ml of the content of each of the container, but not than less than 1 ml has to be used. Greater than 40 ml, not greater than 100 ml, 20 ml of the samples. Greater than 100 ml, if the quantity in that particular sample, if it is more than 100 ml, 10% of the content of the container, but not less than 20 ml has to be used. So likewise, they have given how much for antibiotic liquids, and for solids also they have given, for example, if it is less than 50, this particular pharmaceutical products, you all know, they could be semi-solid, it could be in the form of gels, or it could be in the form of liquid, or it could be in the form of powders, etc. So if they are in the form of liquid, they have expressed in the form of ml. If they are in the form of solids, it is being expressed in the form of grams or milligrams. So for example, solids, if the sample is having less than 50 milligrams, the whole content in that entire, each container has to be used. If it is 50 milligrams or more than that, but less than 300 milligrams, the half the content of the each container, but less than 50 milligrams has to be used. I think you understood with respect to this. So here the main aim of studying this particular table is, the previous table, what we studied was the number of samples out of that particular batch, whatever you have got, either injectables or the powders or whatever it is, after that particular batch, whatever you have got, how much of the samples has to be collected for analysis. That is what we studied in our previous table. So here we are analyzing how many in that particular batch, in that particular sample, whatever you have collected, in that how much ml or how much grams or how much milligrams of that particular sample has to be used for inoculation or for analysis. That is what this particular table tells. So as I was mentioning, if it is in the form of solids less than 50 milligrams, the whole content of the container has to be used. If it is 50 milligrams or more, but less than 300 milligrams of the container of each has to be used, of the content of each container has to be used, but less than 50 milligrams. So if it is greater than 5 grams, for example, if that container contain around 5 grams of some powder or whatever it is, so out of that 500 milligrams has to be used. Then only our analysis, whatever we are going to isolate the microorganism, is correct or not. Likewise, for surgical sutures, that is catgut. So 3 sections of the strands, that is 30 centimeter long of that particular sutures has to be inoculated into the culture media. Likewise, they have also given for surgical dressing, cotton, gauze in a packet. 100 milligrams of that particular surgical dressing or cotton or gauze in a packet has to be taken, 100 milligrams has to be weighed, and it has to be inoculated into the media. This is about what we studied. We studied about how much amount of the sample has to be used for our analysis. One thing what we studied was the number of samples which we have to collect, in that bulk or in that particular batch, and we out of those items, whatever we have collected, this many numbers or this many percentage of items if we have collected, how much quantity, if it is liquid, how much ml we have to use, if it is a solid, how much grams it has to be used, that we have studied in this particular slide. This is also according to USP 71. Okay, so all these slides, what we are studying is, we are studying about how this sterile testing is being done for pharmaceutical products, and we are studying how we have to monitor the working environment, and also what are the things we require for this particular sterile testing. One thing is, first we have to know, once we get the samples in bulk, first you see to that your room, wherever you are going to work, the place we are working is devoid of microorganisms. Then you have to see to that you select the samples, the surface, sterilize those samples, then it has to be brought, that also has to be analyzed, and you have to bring it into your clean room and select the samples. How many samples based upon the number of batch, the number of container in that particular batch, and based upon the amount in each of that particular container, use this USP 71 or Indian pharmacopoeia or European pharmacopoeia. You can use as per the industrial requirement. Use it and keep the things ready. So collect the samples and keep it ready. Now, next. So now our sample is ready. How much number of sample is ready is required? That is ready now. Now, I was talking that use 1 ml of the sample, use 1 milligrams of the sample, 100 milligrams of the sample. Where you are going to use, how do you are going to analyze this? How do you see the microbes present or not? For that, we require what? Culture media is required. For culturing of any microorganism, you require, the next thing what you require is the culture media. So what is the meaning of this culture media? What is culture media? What is culture media?
The nutrition used to grow the organisms.
Yes. So this culture media is, if you are going to grow this microorganism in laboratory condition, we have to provide them some nutrients. So that nutrient is called as, the nutrient whatever you are going to provide them to grow, that is called as culture media. I think culture media, you all know, it can be classified based upon its consistency into how do you classify the culture media? Anybody? Priya and Priya?
That may be a selective media or differential media.
Before that?
A common media, that may be
Based upon the consistency of the media, you can classify them as solid media.
solid liquid
Yes, ma'am.
or
Solid media,
or liquid media.
liquid media, semi-solid media, isn't it? Solid media, why it is solid? Why the media will be solid?
It will be in a solid form.
Why it will be solid?
Uh,
For example, you spoke about selective media, differential media. Can you give some example?
For selective media, only the microorganisms which are
Any example? Any media example? Differential media? Any example?
I don't remember.
Any media? Can you name any media? Any culture media? Name of one media.
Agar.
Agar is one of the component of the media. Any nutrient agar, you know about nutrient agar? Do you know about nutrient agar? Potato dextrose agar, blood agar, chocolate agar. Why it is called agar? Because anyone else, you can classify the culture media based upon its consistency, the concentration of solidifying agent which we are going to use, isn't it? On what basis, how do you classify them, Lasya? What is the difference between nutrient agar and nutrient broth?
The concentration of agar in nutrient solid media, the concentration nutrient agar will be more, so that it will
More in
help
the
in
sense?
solidification. In
Okay.
nutrient broth, we will not be adding this agar for the
Mm.
solidification thing. We
Yes.
need it in liquid form.
Liquid. So you can basically the culture media can be classified as solid media, semi-solid, and the broth. You are calling the liquid media as a broth. So the broth is devoid of any solidifying agent. Agar is lacking. Agar is one of the ingredient in the culture media which makes this particular media solid because it is a solidifying agent. Usually the concentration of this agar is around 1.5 to 1.8%. 1.8% of agar, if you are going to add, the media at room temperature becomes solid. So if the agar, the solidifying agent, if we are not going to add, such kind of media is called as the broth or the liquid media. So semi-solid, if it is around 0.2% or 0.3% of agar, if you are going to add, that is called as semi-solid agar. Okay, so this is about the classification of culture media based upon the consistency, whether it is a liquid or a solid or a semi-solid media. So you can also classify this culture media based upon the ingredients which we are going to use, ingredients, that is the chemicals which we are going to use for the preparation of this culture media. You can classify them as natural media or a synthetic media or a semi-synthetic media. So this natural media, what is the meaning of natural media? So you can classify them as natural, synthetic, semi-synthetic. As the name indicates, natural media, you are going to use some natural resources for the preparation. Synthetic media, here it is being made up of only chemicals, that is called as synthetic media. You have to know this, being the microbiologist, you have to know about the basics I am going to tell. So next, you can also classify this culture media based upon purpose of use. For what purpose we are going to use this culture media, that is also been important. As it was told, this particular media can be a enriched media, as she was telling, there can be a selective media, or it could be a differential media, etc. So what is the meaning of differential media? So as the name indicates, differential media is for example, McConkey agar. Have you heard about McConkey agar? McConkey agar, blood agar. Blood agar,
Yes, ma'am.
okay,
Yes,
McConkey
ma'am.
agar, it's one of a differential media which can be used to distinguish lactose fermenters and non-lactose fermenters, because one of the ingredient we are going to use is the lactose, and we are going to use few of the indicators. Because of these indicators, the color of the colony, if they are red, they are lactose fermenters. If they are not, they are non-lactose fermenters. When you are going to analyze, or if you want to place this bacteria into different genus and the species, first and foremost thing, what we are going to do is whether the particular organism is a gram-positive organism or a gram-negative organism. You know about this gram-positive and gram-negative bacteria. If they are gram-negative bacteria, first and foremost thing, what we are going to do is we will analyze whether it is having an ability to ferment lactose or not. Okay, so for that, what we are going to do, we are going to directly inoculate onto the McConkey agar and see whether it is a lactose or non-lactose fermenter. So if they are producing pink colored colony, they are lactose fermenters, if not lactose, so that it is easy to distinguish or group the organism. So that is the reason it is called as differential media. So such kind of media which is used to differentiate or distinguish two groups of bacteria, they are called as differential media. Then you have a selective media. What is the meaning of this selective media? This is a media which is used to select a particular bacteria. That is called as selective media. So for example, if I ask you to isolate rhizobium, for example, rhizobium. Have you heard about rhizobium? It's one of a symbiotic bacteria, soil bacteria. If I want only rhizobium, if I want only E. coli, what I will do, if I require only mycobacterium, if I ask you to analyze whether, see whether that mycobacterium is present in this sample or not. So what you will do, for example, mycobacterium LJ media, you are going to prepare and you are going to inoculate and see. So that is a media which will select only that particular bacteria. That is called as selective. If I want rhizobium, what I will do? Yeast extract, mannitol, agar I will prepare. If I want Staphylococcus aureus, I will go directly to the Baird-Parker agar. Baird-Parker agar, it is one of the media which can isolate only the Staphylococcus aureus. And looking to the color of the colony, I can analyze whether it is Staphylococcus or not. That is called as what? Selective media. Enriched media. Enriched media in the sense the media in which we are going to enrich by adding special substance. That's called as enriched. For example, blood agar. Have you heard about blood agar? I think you know about blood agar. Why it is called as
Yes,
blood
ma'am.
agar? Why it is called blood agar? Yes, Vaishnu Priya.
Uh, Ma'am,
Correct.
enrichment,
Yes.
liquid medium will be this.
Okay, you are going to enrich the media. This is just a nutrient agar. Nutrient agar that is having nutrient agar. Can anybody tell me the composition of the nutrient agar? Peptone, beef extract,
Peptone,
and
Peptone,
beef
beef
NaCl.
extract, and
NaCl.
NaCl.
These are the ingredients. Into that, you are going to add 5% of sheep blood. Okay, so you are going to enrich that particular blood into this particular nutrient. That is the blood agar. So even this blood agar is also a differential agar, differential media. So here, the hemolysis happen. Why we are going to provide the blood? For the non-hemolytic organism. So this hemolysis can be of three kinds: half alpha, beta, and gamma. Beta is complete hemolysis, alpha is partial, and gamma is no hemolysis at all. Okay, so here, based on that, this blood agar also help us to distinguish that particular bacteria is a hemolytic, complete hemolytic bacteria, or a partial, or it is not a hemolytic. That is the reason it is a differential media. Okay, so here my aim was just to tell what is this culture media. Culture media is, as you all said, culture media is some preparation containing the nutrients which we are going to provide for the microorganism in vitro condition for their growth and development. That is called as culture media. There are n number of culture media. For each of the organism, to each of the species, you can go for the culture media. But according our year, we need not have to use all the different culture media in our sterility testing. So according to USP, we are going to use these three media. Please make a note of this particular media. One is fluid thioglycolate media. Fluid, it is commonly called as FTM, fluid thioglycolate media, alternative thioglycolate media or thioglycolate broth, and soya bean casein digest media, tryptone soya bean broth or casein soya bean broth. So here we are going to use, most of the time we are going to use the broth and see for the growth of the media. See, for example, if you are going to another thing which I would like to emphasize over here is, if you are going to use a solid media, if you are going to use a broth, how do you distinguish the microorganism has grown or not? How do you see that? Can anybody let me know? How do you see the microorganism? How do you analyze? How do you conclude the microorganism is if it is a broth preparation, or if it is a solid preparation? Solid preparation is, that is, your culture media is a solid media. How do you analyze this? I mean, you have inoculated some organism. How do you analyze whether it has grown or not? Whether the
Turbidity,
microorganism,
ma'am.
huh?
Turbidity. In broth, there will
Very
be
good, very
turbidity.
good, very good. If there is a turbidity, indicates, very good, indicates that there is a growth of microorganism. If it is a solid media, if I ask you to inoculate onto the solid media, what you will do? How do you analyze?
Colony formation.
Very good. Colony formation can be analyzed. So here, according to USP 71, these are the media which we can use. One is fluid thioglycolate medium. It is used with clear fluid products, and it is mainly intended to culture anaerobic bacteria, and even we can also use for aerobic bacteria also. So now the question comes, what is the meaning of aerobic bacteria and anaerobic bacteria? So here, kindly note down, we can use, basically according to USP 71, we can use two media. One is FTM, that is fluid thioglycolate media, or you can use any of this soybean casein digest media or tryptone soya broth. These are mainly for fungi. So here fluid thioglycolate media is basically used for anaerobic bacteria and aerobic bacteria. There is alternative thioglycolate media where here it is basically for anaerobic condition, that is for anaerobic microorganism. Soybean casein digest media, it is used for culturing both fungi and aerobic bacteria. So here we have to, the pH adjustment is very much important. One more thing I would like to emphasize over here is, especially for the growth of fungi, always the pH should be slightly acidic. That is around 6.5 or something. If it is bacterial isolation, always the pH is 7. The incubation time for incubation temperature for this particular bacteria is around 37 degrees Celsius. For fungi, it is around 25 to 30 degrees Celsius is the optimum temperature. And here we are, the media, we were talking about the different media. We have FTM, we have ATM, we have SCDM. These are the media which is being approved by according to USP 71. These are the media. They are basically used for the isolation of aerobic bacteria, anaerobic bacteria, as well as for the isolation of fungi. So now let us see. So before that, what is aerobic bacteria? What is aerobic bacteria?
Organisms that grow under the utilization of oxygen.
Oxygen. Anaerobes?
Grows in the absence of oxygen.
Without oxygen, how actually this the metabolism happen there in anaerobic condition? So this is also your assignment. I think we will be having some discussions class now. Presentation class, I will definitely ask about Whittaker system of classification, aerobic, the difference between aerobic and anaerobic microorganism, the different culture media, you have to thoroughly, you should be knowing. So according to this USP, definitely these are the only two media, but I will, to make you a proper microbiologist, with respect to this sterile testing thing, I want you all to understand the different media, the composition of the media, the principle behind those media. I request you all to know that. Definitely, even though it is not there here in my MCQs, those questions I'm going to ask, and even during the presentation, you have to answer me those questions, students. Please make a note. What are the things? Whittaker system of classification. Whittaker system of classification, the difference between the aerobic and anaerobic microorganisms, and the culture media, isolation techniques, isolation of bacteria and fungi, the different techniques. These are the basic things you would have studied in your UG itself that we are going to discuss. So now we understood how the sampling has to be done according to USP 71, how many samples has to be collected, how much amount of sample has to be collected, and we started with the study of this culture media. The important culture media according to USP 71, we are going to use FTM, that is fluid thioglycolate medium, and we are going to use one more media called as soybean casein digest media, which is especially used for the growth of what? Fungi. So here in this particular slide, I have given the composition of the culture media. Composition, please make a note of this composition. The first column is fluid thioglycolate media. Second column, FTM is fluid thioglycolate media. ATM, alternative thioglycolate media. And the third last media, that is soybean casein digest media. So here I have given the composition for each of the media. This is also important. So you kindly make a note. This could also be asked as a MCQs in my test. Please make a note. I request, I think everybody have your notepad, pen and all now. Notes, please make a note. Casein, you have sodium chloride, dextrose, yeast extract, pancreatic digest, sodium thioglycolate, Rizar's urine, distilled water. This is the composition. This is the composition for 1 liter of the media. 1 liter of the media. The main purpose of adding yeast extract, pancreatic digest, dextrose, cysteine and all, cysteine, usually for the carbohydrate source. You have sodium thioglycolate, which is important. The main purpose of adding this is, please make a note, it maintains the reducing condition in the media. Main purpose, why we are adding the sodium thioglycolate, thioglycolic acid, maintain the reducing condition and also protect the enzyme against inactivation by maintaining the protein thiol group in reduced state. So mainly it helps in maintaining the reducing condition and also it protects the enzyme against inactivation. How it will protect the enzyme against inactivation? By maintaining the protein thiol. Thiol means what? Sulfur group in the reduced state. Rizar's urine, sodium soluble, 1%, 0.1% you are adding. Rizar's urine, we are going to use for the preparation of fluid thioglycolate media. Especially, this is one of the media which is used for the culturing of anaerobic bacteria. The main purpose of adding the Rizar's urine is, Rizar's urine will be bluish in color. On oxidation, they become pink in color. So if there is no color change, indicates if it is turbid, if the media is turbid, indicates that the anaerobic organism is grown. So in case of ATM, there is no Rizar's urine. the turbidity can be seen. So here towards the right, I have given the uses, where this particular medium have been used. Fluid thioglycolate media is used for the growth of anaerobic bacteria. For sterility testing, and you have soybean casein agar digest media, that is trypticase soy broth suitable for culturing of fungi and anaerobic bacteria. Please, all of you, remember these media. What is the composition of the media and where it is applicable? That is important. So I'm making you to write this composition so that at least you will remember. That is the reason. Have you finished? So how do you prepare this media? For example, if I ask you to prepare FDM, prepare. So nowadays, usually in companies and all, they have such kind of ready-made media are available. You need not have to prepare. Ready-made media, sterile media are available. Into those media, our test organism or our test sample has to be inoculated. Okay. So if you should also know how it has to be prepared. You may tell that those are available, just we are going to add. It is not the case. We should know how it has to be prepared. Here, the composition has been given. This is for 1000 ml. Weigh all this ingredient and dissolve in 1000 ml and set the pH for 7.1 to more or less 0.2 difference. Sterilize in an autoclave. You know what is autoclave? That is one of the method of
Sterilization.
Yes, 1 second. 1 second. My here the laptop battery is low. I should shift something else. 1 second. Uh, 1 second. Sorry for the inconvenience. Okay. Okay. Sorry. Can you all hear me? Can you all hear me? Shashank?
Yes, ma'am.
Yes, ma'am.
Okay. Okay. So weigh all these ingredients and dissolve in the 1000 ml of water, set the pH and sterilize. Usually the sterilization is done by the process of through autoclave. Autoclave is the moist heat method of sterilization. What is the temperature we are going to follow? What temperature the sterilization is been done there in autoclave? Shubhadeep? Shubhadeep?
Yes, ma'am. Steam
Yes, ma'am.
sterilizer.
Yes, ma'am. At what temperature?
121
Very
degrees centigrade.
nice. Very nice.
At
How long?
15,
Very good. 15?
15
Uh,
minutes.
and it is under pressure, isn't it?
15 psi.
PSI. What is PSI?
Um, I
Pounds,
forgot.
yes, pounds per square inch. Okay,
Okay.
at that particular temperature for 15 minutes, at that particular pressure, if you are going to sterilize, they are devoid of microorganism. Such media has to be kept ready for our what? Test. Okay, so once the media is ready, so as I was mentioning, so the media, the readily available media are also there. The media you people can also prepare, but first, before going for inoculation, we have to validate the media, whether the media, whatever we have prepared or whatever we have got, whether that media is working or not. That for that reason, we have to go for growth promotion test. What is this growth promotion test? By where we are doing this? Growth promotion test. For what we are doing this growth promotion test? So now our media is ready. Media has been sterilized. Now we have to analyze, we have to validate that particular culture media is proper or not. In simple words, that is called as validation. For that, we have to perform growth promotion test. So here it is, this is the main purpose of this growth promotion test is to test the ability of the media to support the growth of microorganism. Okay. So what is this growth promotion test? To see whether, so you have prepared this media or you have obtained such kind of media. Okay. So now in your clean room, imagine that you are in clean room, you are monitored your clean room, you have collected the sample and everything is ready and your culture media is also there. Culture media, whatever you are taking, it should be validated. Whether it is suitable for use, also we should understand. For that, we have to perform this growth promotion test. That is the ability of this particular media to support the growth of microorganism. Media, that is, it should be inoculated with 100 CFU of challenge. That is the challenge organism. Our organism, I will talk about which are the organisms we have to inoculate. According to USP, there are the list of organisms which we have to use to see whether this particular media, it could be validated or not. So for that, 100 CFU challenge organism has to be inoculated. So once the media is media, you have inoculate the test organism. Test organisms, these are the test organisms. 1 second. Yes. According to, you have to remember these organisms. You have to remember these organisms. Please make a note. So what I am talking over here is growth promotion test has to be performed. How do you perform this growth promotion test? Whatever the culture media you have received, in order to see whether those culture media is proper or not, in order to validate this culture media, we have to inoculate few test organisms and see, yes, this particular media is appropriate. How we are inoculating, how much we are inoculating? It is 100 CFU, more than 100 CFU. What is the meaning of CFU? Colony forming unit. That is the growth condition. So much microorganisms has to be inoculated into the culture media, incubated, and we have to see, analyze them after 3 days of incubation. If there is a growth of bacteria, after 5 days of incubation, if there is a growth of fungi, indicates that this particular media is suitable for usage. Understood? Understood. What is this growth promotion test? This is to validate whether the culture media which we are going to use is appropriate or not. So for that, what we are doing, we are inoculating the test organisms. Which are those test organisms? it could be it is a combination of 3 different kinds of organism. One is aerobic bacteria, anaerobic bacteria, and fungi. What are these aerobic bacteria? As you all know, as it was told, they are the group of organisms which require oxygen for their metabolism, which anaerobes, they doesn't require this oxygen for their metabolism. And the fungi, these group, according to USP, these are the organism which can be used. And also these could be, also they have given the code also ATCC. What is the meaning of this ATCC? America, these are the culture collection centers. ATCC, and that is the strain. ATCC 6538, that is the strain of Staphylococcus aureus. Remember. Okay, apart from remembering the genus and the species, remember the strain, the ATCC 6538. CIP, what is the meaning of CIP? Everything I have given there down. What is NCTC? Everything, National Collection of Type Cultures. These are the collection center, and the number is being given for Staphylococcus. ATCC 6538 is the strain of, that is the strain of Staphylococcus under the collection center of American type culture collection. So that also you have to remember. For these strains, only for these group of organism, according to USP 71, we are going to use this as a challenge organism or the test organism to validate our culture media is suitable for the usage for testing the for performing the sterility test or not. Staphylococcus aureus, Bacillus subtilis, Pseudomonas aeruginosa, these are some of the aerobic organisms which you are going to test, whether those can grow in our media which we have prepared. Clostridium sporogens, one of the anaerobic bacteria we are going to analyze for its growth in anaerobic media. Likewise, we have Candida albicans, that is the representative of yeast, and Aspergillus niger, the representative of molds. So these are our challenge organism, where we are going to use this challenge organism for growth promotion test. Okay, so you are going to prepare these broths. You are going to prepare these broths, and to see or broths you would have obtained to validate this culture media. We are going to perform growth promotion test. Growth promotion test. To perform this growth promotion test, we are going to inoculate these organisms separately, not all together, separately into this culture media, and in that particular and see whether that particular culture media could be validated or not. One thing is whether this particular media support the growth of microorganism. Another point of thing is whether that culture media which we are going to use is sterile or not. Sterile in the sense that media, whatever we are going to use for testing, whether it is devoid of microorganism. That is, that also we are going to analyze whether after the period of incubation of that particular media, after 14 days of incubation also at 30 to 35 degrees Celsius, if there is no growth, indicates that media is sterile. We are conducting 2 tests. Don't get confused with both of the tests. One thing is to see whether the organism is growing. That is different test. We are going to inoculate these organisms and see whether those organisms could grow in this media. If it grows, fine. This media could be validated. Another point of view is we should see whether that particular media is sterile or not. Sterile in the sense it is devoid of microorganism. So for that, we are going to incubate this media for 14 days at appropriate temperature for fungi and bacterial growth, and see if there is no growth, no turbidity or nothing grows, indicates that the media is sterile. If it passes both the test, indicates that our culture media, the culture media could be validated. Okay, so now the culture media is also ready. Sometimes what happened is, if you want to analyze penicillin, for example, some antibiotics, if you want to analyze to see whether the microorganism, you want to see whether microbiological analysis or the sterility test if you have to conduct for some antibiotics, so for example, penicillin, cephalosporin, streptomycin, many other components. So what we are going to do, since it itself is an antibiotic, inhibits the growth of certain bacteria. So what we have to do is, we can add, for example, penicillin is the antibiotic which we want to test for sterility testing. So what we can do is, we can add penicillinase into that particular media. So it inactivates the penicillin, and we can see whether the bacteria is growing or not in that particular media. If it is cephalosporin, the antibiotic solution or the powder, whatever it is, if we have received for quality control or to analyze the microbes, to analyze its sterility. So if cephalosporin itself is antibiotic, if you would like to see whether the microorganism is there or not, what we have to do is, cephalosporinase has to be added into the broth culture media. So it inactivates the cephalosporin, and then it will promote the growth of bacteria if it is there in that particular thing. Okay, so this is the list. This also you have to remember. This is some of the antimicrobials. If we have to analyze, I have given the list of inactivating them. Antimicrobial agent, if you want to analyze its sterility testing has to be done for antimicrobial agents. Those has to be inactivated. How we are going to inactivate them? By adding appropriate substance or inactivating agent into the broth or into the culture media, inactivate that particular antimicrobial agent and promote the growth of microorganism if it is present in that particular media or not. Okay, so this also very much important, you have to remember. So now, so how do you perform these tests? So now, once again, our room is ready. The room is wherever we are going to perform these tests, sterility test. Our place of working is clean. It is sterile. The environment is sterile, and the person who is going to handle that, even he is also devoid of the microorganism on the surface of the wearing, whatever he is going to wear. And the sample is ready. This many number of sample you have chosen, and what amount of sample has to be used, that also you have taken into consideration. Your culture media is also validated. Now you have to inoculate, isn't it? Now, whatever the pharmaceutical products have been given to you, whatever you have chosen as a sample, whatever the amount of sample you have chosen, that has to be inoculated. So how this inoculation can be performed? There are two different methods of testing according to USP 71. One is membrane filtration method, and the other one is direct inoculation method. Whatever the sample you have received, you can analyze them. Analysis of or the isolation of the microorganism can be done by membrane filtration method. The other one is by directly you are going to inoculate the sample into the media. So first, let us study about membrane filtration method. I think this is membrane filtration. Membrane filters, you know, this is one of the method where this is also one of the method of sterilization, where especially for antibiotics or some serum samples, if you want to sterilize them, because you cannot autoclave them, you cannot go for dry heat method or pasteurization method. We can go for filtration technique. There we are going to use this filters. Same kind of filters can be used here. The main purpose of using this membrane filters is to trap the microorganism which is present in our sample. Okay. So for example, if this is your sample, you are going to pass your sample through this membrane filtration disc, and you are going to collect the filtrate. This filtrate is sterile. The microorganism, if it is there in that particular sample, that is going to get trapped into this filter. That is the main principle over here. Okay, so here membrane filters, usually the nominal pore size is around 0.45 microns. Remember, please make a note, the not greater than 0.45 microns. What is microns? That also important. You should know what are microns. 0.45 microns. They are very minute pores. The bacteria or the virus, bacteria could not, or any fungi or any of the microorganism could not pass through this particular membrane. So according to USP standard, the filter pore size, we are talking about pore size, not greater than 0.45 micron. So what are these filters? Here I am pointing out these filter I am talking about. You are going to pass the sample through this filter and you are going to collect the sample. Sample is not our target. The membrane is our target here. So if the microorganisms are present in that particular sample, they are getting trapped in this particular membrane, and that membrane we are going to use for culturing. Okay, the microorganism present or trapped in that particular membrane, that we are going to culture. For that, the pore size of this particular membrane, which we are going to use, is 0.5 micrometer. And the diameter of this particular membrane filter is 50 millimeter. Remember, it is 50 millimeter. Diameter of this membrane is 50 mm according to US. Flow rate in the sense the flow rate, the volume of this particular sample passing through this is flow, that is called as flow rate, that is 55 to 75 ml per minute. In 1 minute, it can filter, the solvent or solution can filter only 55 to 75 ml of the test sample can be filtered per minute at the pressure 70 mm Hg. This particular filter is usually made up of cellulose nitrate. Okay, so this is what the filter is. So once you pass this particular sample through this filter at this particular flow rate, and so the microorganism get trapped over here, and such filter you are going to collect, take out back with the help of sterile forceps, take it back, and then you can make it into two halves. You are going to make it into two parts. One part you can inoculate into which media? FTM. The other FTM or ATM. FTM is preferable because both aerobic and anaerobic organism grow here, and SCDM, that is the media which is used for seeing whether the fungi is being grown or not. So take that particular filter aseptically, cut it into two halves and inoculate into the two different media. What is FTM? FTM is for bacteria, SCDM is for fungi. And once you have inoculated in such kind of media, see, this is how the media will be. See, such kind of bottles are available so that you can directly inoculate into this media, and you are going to incubate at 20 to 25 degree or 30 and fungi 30 to 35 degrees Celsius. Incubate for 14 days and see for the turbidity. Okay, so this membrane filtration technique is being used for the oil or oily preparations, soluble powders, liquid products, ointments, etc. Okay, so this is about membrane filter. Membrane filtration method are of two kinds, as I was mentioning. One is open membrane filtration. The other one is sorry. So it could be, as I was mentioning, it could be of open membrane filtration and closed membrane filtration. Can you hear me? Can you hear me?
Yes, ma'am.
Yes, ma'am.
Okay, sorry.
Yes,
Okay.
ma'am.
Okay. There was some internet issue here. That's the reason. So open membrane filtration, as you could see in this particular image, you are going to filter this, say for example, this is the membrane filter and these are the membrane filtration unit. These are the membrane filtration unit. This is being connected to vacuum pump to have such kind of pressure. So you are going to take out this particular, the membrane filter, you are going to place it over here. And through this, you can pass the sample and later you can take out this particular membrane filter, cut into two halves. One you can inoculate it into the media, fluid thioglycolate media, and another one you can inoculate into the fungal growth media, incubate and see for the growth of the organism. That is open membrane filtration system. There is one more thing called as closed membrane filtration technique. So here you need not have to take out those particular filtration discs. Directly you can add, after the process of filtration, you can directly add the media into this. That is the reason it is called as closed membrane. So here what we are going to use, there is something called a syringe-like substance here. So you are going to draw the sample whichever you would like to test. So for example, if you have an ampule of antibiotic, if you have an ampule of distilled water or whatever it is, if you want to take out 1 ml of the sample out of that, you are using this thing, you are going to collect that particular sample and you are going to pass this particular sample through this membrane filtration unit. So it gets filtered. So later what you are going to do is, you are going to add the culture media into this particular system. So that is the reason it is called as closed membrane filtration. So in my previous thing, you are going to take out that particular filter paper, you are going to cut into two halves and you are going to inoculate into the sterile media. Here you are not going to take out, just you are going to add the media into this particular system. That is the reason it is called as closed membrane filtration system. Then other important method is direct inoculation method. Direct inoculation method, very simple. What you are going to do, the test sample, the amount, what is the volume of the sample or the ml in the form of ml or in the form of grams, you are going to take out that particular sample. Directly you are going to, without any filtration or whatever it is, without trapping the microorganism, you are going to directly drop it or inoculate into a culture media for bacteria to see for aerobic, anaerobic or the fungal media. And you are going to incubate for 14 days and you are going to interpret the results based upon the growth or absence of the growth of the media. So this is called as direct inoculation. So which are the different methods which we can follow for the sterility testing according to USP? This is according to USP. There are many more methods which we can follow. This is as per USP. These are the different technique which we have to follow. One is membrane filtration method. Membrane filtration, there are two methods. One is open system method and the other one is closed. And another method is direct inoculation. The sample which you have to analyze is being directly inoculated into the media and you are going to incubate and see for the presence or absence of the growth. So once the incubation is done, you are going to analyze or interpret your test. So as you could see in this particular chart, after incubation and during the incubation period, if the growth is observed, if the growth is not observed, okay, so you have inoculated your test organism, test sample, and you are analyzing, and if there is, after 14 days of you are incubating for 14 days, after 14 days of incubation, if there is no growth you can measure. If there is, here we are going to use broth. So turbidity can be seen. If it is not turbid, it is clear when compared to that of control. Control in the sense uninoculated sample compared to that of if there is no growth. So indicates that it passes the sterility test. Indicates that that particular sample is devoid of microorganism. Okay. So if growth is observed, so what you have to do, whatever the sample you have taken, again you have to go for retest. Again you have to go for retest. If the growth is observed, if they are not distinguished from these growing in container reserved in the first trend, preparation fails for the test. That is, if they are not readily distinguished from in the first test itself, you had got the growth. So here, if there is no growth, that is the preparation fails for the test. If there is a readily distinguished from those of growing in the container reserved in the first test, second retest, again if there is growth, you can again perform the test and analyze if there is not growth observed, the preparation is being passed for test, that is, it is sterile. Again, if there is growth is observed, our product fails for test, that is, indicates that our sample is not sterile. So once again, I will repeat, after the period of incubation, if no growth is observed in first slot itself, that particular batch, that particular whatever the sample, it passes through the sterility test. If growth is observed, you have to go for one more round. Again, you can see whether the growth is there or not. Again, if there is growth, you can go for second round of sampling. Again, there, if the growth has been observed, indicates that the sample, whatever you have received, is not a sterile one. Okay, so this is how we are going to see for the sterility testing. So once again, let us just brief whatever we have learned here. So here we learned with respect to this microbiological analysis, with respect to the sterile manufacturing process, we are concentrating on sterility test. We studied what are these, when you are going to call this as a sterile, what is the meaning of sterilization, why the sterility testing is required. Apart from having the viable microorganism, you could also see some biogens, that is the lipopolysaccharides of the gram-negative bacteria, if it is present in this particular sample or not, that can be analyzed by LAR test or by rabbit pyrogen test. And our main aim is to study the microorganism, viable microorganism in the given sample. For that reason, we have to see environment has to be monitored, where we have to monitor the personnel, we have to monitor the air, we have to monitor the surface of that particular area where we are working. And these are the products, which are the pharmaceutical products we can analyze. And how do we analyze? What are the different steps involved? First, we have to see for the sample size and the volume of the sample, and the media is required, the different media according to USP, the composition of the media, and to validate this particular media, we have to perform growth promotion test, and we have to also see whether this media is sterile or not. Once it passes through this test, that indicates that the media is validated. And to perform this validity test, these are the microorganisms which we are going to use, which includes aerobic bacterium, anaerobic bacterium, few of the fungi, which representative of yeast is Candida albicans, representative of Aspergillus, molds Aspergillus niger we are going to use. These are the collection centers through which we can procure them. And also, sometimes the antibiotics also we have to analyze in order to deactivate them, and inactivating agents has to be added into this media. These are some of the examples I have quoted over here. And the techniques which we have to follow here is membrane filtration technique. We have open membrane filtration technique as well as the closed one, and the other one is the direct inoculation method. And finally, once based on the presence or absence of the growth, we can interpret the result and give the report. So this is about sterility testing of the pharmaceutical products. Okay, students, any doubts here?
Ma'am, I have a doubt.
Yes, ma'am.
Ma'am, the culture media,
Mm.
FTM, it is for bacteria and SCDM for fungi.
Mm.
What about the ATM, ma'am? Whether it is for bacteria only?
Well, that is also for bacteria.
Not for
This is also for, no. This is for not for aerobic bacteria.
Okay, ma'am. One more doubt, ma'am, regarding the strains.
Uh-huh.
There is a numbers, right? ATCC.
ATCC, that is the culture collection center. If you ask for ATCC 6538, Staphylococcus, that's strain.
For each one, it will be different number, ma'am. Strains.
Yes, ma'am. Yes, ma'am. Yes, ma'am. That is the culture collection center has given that particular code for that particular organism.
Okay, ma'am.
That particular strain has to be used for validating the culture media.
Thank you, ma'am.
So,
If
student,
you have any doubt, you can clarify.
prepare. So prepare well for your MCQs and even for our discussions. I have given few of the topics. Apart from these, kindly let us have a discussions on those basic microbiological techniques.
If nobody have any doubt, please we'll stop for today's session. And so thank you, ma'am.
Okay, thank you, Vishal. Thank you, Vishal.
ma'am.
Thank you so much, students, for your
Thank you, ma'am.
Thank you, ma'am.
interactions.
Thank you, ma'am.
Thank you, ma'am.
Thank
Thank you,
you.
ma'am.
Thank you,
Thank you,
ma'am.
ma'am.
Thank you so much. Thank you.
Thank-
Any
thank you,
doubts?
ma'am.
If you have any doubts, you can message me also. Okay, thank you. Thank you a lot.
Thank you, ma'am.
We all meet not next week, I guess, for our next session. Okay. Thank you.